Okay, so the first principle of cell theory, all living things are made of cells. Second principle or tenet of cell theory, the cell is the basic unit of life. Do you need to know those two? Absolutely. Did we talk about the different levels of organization of life? Yes, no, maybe? Cell is the most basic unit. We work our way up from that. Sure. The cell is the basic unit of life. When I say the basic unit, I mean there is nothing smaller than a cell that is considered living. Your body has probably on the order of 50 billion cells. That's a lot. But every single one of you started life as a single cell. About 30 minutes of your existence on this mud ball were spent pretty much the same way an amoeba does. Kind of puts it in a little perspective, doesn't it? The main thing I want you to understand here, though, if you don't understand cell biology, you don't understand life. Now, above that, level 2, we've got tissues. Tissues. There are 3 different types of tissue that we're going to talk about. We'll get to those in the next chapter. Above tissues, and you all want a definition for that? A group of cells with a common function. A group of cells with a common function. Dermal tissue, that's what makes up your skin, for example. Now you got all kinds of things going on in the skin, don't you? You got fingernails, you got hair, you got the skin itself. But the bottom line is, it's a covering. Does everybody see how this works? This is a very, very generalized statement. Above tissues, you now have organs. Organs. Organs are simply a group of tissues with a common function.
Can you repeat that again?
I sure can. They are a group of tissues with a common function. Example of that, consider, let's say, the small intestine. It's lined with dermal tissue, but it also is lined with smooth muscle tissue, which kind of squeezes and moves the digestive process along. It's lined with blood vessels that take up the food. You see how this goes? That's an organ. The next level above that would be organ systems. Organ systems. A group of organs with a common function. The digestive system. You've got an entrance, you've got an exit, you've got a whole series of organs that are involved from start to finish. Esophagus, stomach, small intestine, large intestine, pancreas, liver, you get the idea. And then finally, the final level for us would be the whole organism, the whole body. And I think that's pretty self-explanatory. And that's when I talk about the organization of life, the different levels of life, that's what I mean. Is everybody okay with that, comfortable with that? You don't have to write down what I'm gonna say next, so I just want you to be aware. You can extend that further. For example, a population. That's just a group of organisms in the same place at the same time. Are we a population here? Yes, we are. We have different interactions with each other, don't we? We could expand that to say the entire campus. Then we could go with the idea of a community. And a community is just a different group of organisms interacting. If we expanded it to the whole campus, including the environmental thing there, then we've got humans, don't we? We've got the various viruses and bacteria that we're carrying around, don't we? We got the birds out there landing on the lawn. We've got the trees, the grass. You see where I'm going with this? And you can keep going up some. We're not going to worry about that. This isn't an ecology class. I just want you to understand, we as humans like to organize. We put things together in categories so we can study them. We can also go below that. What's below the cell? Molecules. Molecules. And then below that, we've got atoms. Everybody see how this goes? But you can't call molecules alive, you can't call atoms alive. The only thing you can say that's living is a cell. The only thing you can say that is actually living is a cell. The cell itself is made up of a lot of different structures. Let's talk about some of these. The most basic classes of molecules. The most basic classes of molecules. Number one are carbohydrates. Carbohydrates. These are primarily for short-term energy storage. Short-term energy storage. We've all been there. You get tired, you eat a candy bar, you perk up a little bit, right? You get that burst of energy. The reason we say short-term is because you break them down pretty quickly. You don't get as much bang for your buck, but they're cheap and plentiful, and you get them quickly. That's the key. They also play a structural role. Make sure you get that down. In humans, we don't really consider the structural role as much, but it is important because, anybody here ever heard of fiber in your diet? Roughage? That is actually a form of carbohydrates from plants which we cannot use. Most of you got a sheet of paper in front of you. Paper is made up primarily of cellulose, isn't it? Anybody in here ever snapped a celery stick? Shredded a leaf? All of those are cellulose. Cellulose is a structural carbohydrate in plants. Humans cannot break it down. It literally passes right through your digestive system. So carbs are basically structural and short-term energy. Short-term energy. Question?
No.
Everybody good? Okay. Second up, lipids, lipids. Basically, in humans, that's fats. Technically, it also includes things like oils and waxes, but the only difference between an oil and a fat is whether or not it's liquid or solid at room temperature. Waxes are just specialized fats that help in waterproofing. Main function in humans, number one, long-term energy storage. The thing to remember here is lipids give you more bang for your buck. They store more energy per unit of mass than carbohydrates do. In other words, if you burned a kilogram of carbohydrates, you're going to get less energy than if you burn a kilo of fat. Everybody see how that goes? It's ideal for long term. The problem is you don't get that energy quickly. Most of us have probably skipped a meal at some point, am I right? Some of you may have done it this morning for all I know. You get hungry, but your body has a short-term reservoir of something called glycogen in the liver. And as it breaks down the glycogen, it converts it to glucose, and you've got your short-term fuel. It's one of the reasons why you can skip a meal without really, really crashing hard. The problem comes long-term starvation. I told you all about my illness. When I was in the hospital for 5 weeks, I lost 54 pounds. Good thing I had a little bit of extra at the time, because that was the long term. I basically just could not eat. But since I had fat reserves, my body was able to break that down over that period and keep me alive. Is everybody cool with that? The second role that it plays is insulation. Insulation. These people that like to climb Mount Everest, have you ever seen them? They're using the bottled oxygen, and they're wearing their electrically heated parkas and got all the $50,000 worth of equipment and whatnot. And it's still a dangerous thing, isn't it? So like 1 out of 10 people that go up there die. Excuse me. Sorry about that. Most of those people, if you look at them, are big, my size. All of them, the people who are not native to Nepal, have to have the Sherpa guides to help them out, don't they? You ever seen the Sherpas? They're very, very compact. They have a lot of really wired muscle and a layer of fat that insulates them, because the Himalayas and mountains are very cold, aren't they? People who live in colder areas tend to be shorter, more compact. Third role is cushioning. Cushioning. Your organs have a built-in cushion to a degree. It's more effective than you might realize. Is everybody okay with lipids? Third, we've got proteins. Proteins. Proteins play a couple of roles. Their number one, and by far most important role, is catalytic. Catalytic. Now, what do I mean by catalytic? If I asked you what a catalyst was, some of you might say it's something that speeds up a reaction. I think we've heard that in chemistry, haven't we? But how does it do that? Well, the answer is it lowers the energy of activation. It lowers the energy of activation. Now, what do I mean by that? Piece of paper here. We all agree that's what this is, right? Could I get this piece of paper to ignite and start burning?
You had a lighter?
Huh? Who said
Good
something?
with a lighter?
I could with a lighter, thank you. The rest of y'all begin to worry. I'm like, do y'all think I'm Superman? Is he the one that's got the heat rays? I can't just cause it to burst into flame, right? I have to apply energy to start the chemical reaction. That lighter that I hold to this is the energy of activation. Does everybody understand that? Thumbs up if you do. Okay, I'm gonna take you at face value. Start your, sorry. The reason this is important, think about what people say about burning sugar or burning fat. You don't have a literal fire burning in your gut with some shrunken down dude with a pile of sugar shoveling it into the furnace, do you? Yet you break that sugar down to get energy. The key there is you're not burning it, you're breaking it down chemically. That's what burning is. It's just an oxidation reaction. Anybody here ever make caramel? How'd you do it?
Wait, what'd you say?
How did you make caramel?
Oh, no, I thought you said try it.
Well, the way to make caramel is to heat sugar on a stove until essentially it browns and melts. It's not hard. Key thing is you don't want to crank it up too hot, but I guarantee you, once you've browned it and melted it, you're not going to stick your finger in there while it's on the stove and lick it, are you? Because if you do, you're going to get burned. You see where I'm going with this? All of the chemical reactions in the body take place at 37 degrees Celsius. That means we have to effectively oxidize sugar at what is essentially body temperature. This occurs because of catalysts, specifically something called enzymes. Enzymes are protein-based catalysts. They lower the energy of activation so that you can, quote, burn things at body temperature.
Can you repeat that?
They lower the energy of activation so that you can effectively, quote, and I put this in air quotes, burn, in other words, break down things at body temperature. If you don't have enzymes, you don't live. Period, bottom line, end of story. A lot of genetic conditions are caused by malformed enzymes. The gene has an error in it, so when protein gets made, it's twisted a little bit. It's missing a key component, and as a result, it either does not work as well as it should, or it does not work at all. Everybody clear on that?
Can you say that one more time?
Which part?
With the genetic conditions and then.
Most of those are due to a malformed or non-functional enzyme. You get a mutation, which is a change in the DNA. The mutation means that the protein is twisted, it's missing a piece, it's short, whatever. Can you imagine if I took a blowtorch to that lock? Is my key going to fit in there? It might get in, but it might not turn, right? We don't know. It may stick. Anybody ever dealt with a stuck lock? You get the idea. Enzymes are just like that. Shape is most important. And I cannot emphasize this enough. If you don't have functional enzymes, you don't live. Proteins also play a secondary role as structural. They tell you if you want to build muscles, eat proteins. That's just putting structure in to your muscle cells. And then the last two, they share a common chemistry, but they could not be more different in function. This is DNA and RNA. DNA has only one function: information storage. That's all it does. Information storage. DNA is incredibly stable. I can tell you stories of how they have solved decades-old crimes because of DNA that was taken off a 50-year-old piece of cloth. I know one guy in England who claims to have solved the Jack the Ripper murders because of that. They have been able to bring back a subspecies of palm tree that was extinct for nearly 2,000 years because they found a basket of burned seeds sitting up on top of a fortress in the Judean desert. That's a story. DNA is incredibly stable. Y'all want to hear a really wild one? I literally read this morning just before I came over here. Y'all know what the moa is? The moa was the bird that was about 7 feet tall that lived on New Zealand. They actually were able to identify that it ate a certain fungus. Now, the moa has been extinct for about 600 or 700 years. You know how they were able to identify the fungus that it ate? They found fossilized poop in a cave and they got DNA from it. That is how stable DNA is. It lasts. But the only thing it does is store information. And I'm not going to go any further than that because even though I'm a geneticist and I love this stuff, we don't want me going that way. Any questions about that? What is the information that it stores? Basically, blueprints. How to make proteins. Those proteins, in turn, are the enzymes and the structural things. Now again, I'm painting with a pretty broad brush here. I'm more or less summarizing the entire field of biochemistry in about 20 or 30 minutes. But we don't need to go into more detail than that, do we? We're not biochemists here. But you need to have a basic understanding how this all fits together. Which leads us to the last of our molecules, and that is RNA. Unlike DNA, RNA is the most unstable thing in existence. One of my old professors used to joke that RNA would fall apart if you looked at it wrong. It wasn't exaggerating. The one time I did RNA work, y'all want to hear this story? I had to do the RNA extraction in a walk-in refrigerator. Literally, one of those big restaurant coolers. Everything had to be done in there because it's only stable at about 4 degrees Celsius. The glassware that I used, I had to wash with acid. Then I had to bake it in an oven for 2 days. And I could only use it once for that particular thing. I was not successful. My advisor laughed and said, "Great, you get to do it again next week." And I laughed and said, "Ha ha ha, very funny. If that's not a joke, you get a new grad student next week because I'll be quitting. No more RNA work." What does RNA do? Pretty much everything. It is the workhorse molecule. It's a messenger. It carries that information that's found in the DNA. It's transport. It brings the amino acids together so they can be assembled. It's structural. It's a component of ribosomes. It can even be catalytic in certain cases. In even rarer cases, it can store information. cases, something like a retrovirus. That's one of the reasons things like HIV is so deadly, because RNA is the genetic information. Unlike DNA, it's unstable as can be, but that's an advantage if you're a virus, isn't it? Because it means you mutate like crazy. It's one of the reasons why viruses are so difficult to treat. It's one of the reasons why certain things can't be cured. Y'all wanna hear a good one? Why can't we cure the common cold? The answer is there's no such thing as the common cold. Did you know that? What we call cold should be called generic upper respiratory viral infection. There are over 100 different viruses that can affect your upper respiratory system, and all of them produce similar symptoms. Didn't know that, did you? That's why I always tell people, the world is shades of gray. It's not black and white. Sometimes you have to go deeper. What do you got, about a page or nothing so far? Okay, so I'm not going too fast, too hard. Make sure you pass it back on what everybody said. So, that's it for the molecular level. Now let's move into the cell itself. Now it's important to understand, when I say the cell's the basic unit of life, I mean just that. We can take smaller structures out of the cell. We can manipulate those structures, those molecules. What they cannot do is survive outside the cell on their own. Is everybody clear on that? They have to be inside the cell to function, to survive. The cell itself is made up of what we call organelles. It literally means little organs. Now these are not free living, they're not alive in the sense of the cell being alive. They're just structures within the cell that function to keep it alive. And let's start in the center and work our way out. Sound good? In the center, you've got the nucleus. The nucleus has one function and one function only. It stores DNA. Imagine if I had only a single copy, physical copy of the textbook. No electronic copies, it's old school. I want everybody to have the information from that. I or you are gonna have to make copies of that master copy, right? Have to run, find a photocopier and run them off like we used to. That's kind of how DNA works. Those paper copies that we hand out, that's ours. They're temporary, right? You're gonna read it, you're gonna use it, take notes. Once the test is done, you're gonna pitch it, you don't need it anymore. But that master copy, I'm not just gonna hand it to any old person. Hey, here, Lydia, you can take it. You need it next week, and Lydia's like, oh, I forgot it at home. It's useless. Does everybody see how I'm going with this? That's why I say the only function of the nucleus is to be the vault to keep that master copy. Surrounding that, you have a network of membranes. These membranes are called the endoplasmic reticulum. The endoplasmic reticulum. Would y'all be okay if we abbreviated that henceforth as capital E, capital R? Every biology class I've ever had ends up abbreviating that as the ER. Doesn't mean emergency room, it means endoplasmic reticulum. The endoplasmic reticulum comes in two types. The rough ER and the smooth ER. The rough ER is studded with structures called ribosomes. Ribosomes. R-I-B-O-S-O-M-E. Ribosomes make proteins. That's all they do. That's the site of protein assembly. It's where the amino acids get put together. The rough ER serves as the site of protein folding and assembly. Protein folding and assembly. When proteins come off the ER, it's more properly called a peptide chain or a polypeptide chain, because it's nothing but a chain of amino acids linked together. Has anybody in here ever seen origami? Do you know what that is? It's paper folding. And I don't mean making a paper airplane. I mean, you give somebody a sheet of paper and they've got little birds flying around in a lake. Yeah, I could not do this. I had a former student and she was so talented, she could basically just visualize something. You could hand her a sheet of paper and 5 minutes later she'd hand it to you. It's a work of art. It literally is a form of art. Protein assembly and folding is like that. It's how you arrange that strand of amino acids into a three-dimensional structure. It's how you put it together. All of that occurs in the rough ER. Everybody with me? The smooth ER lacks ribosomes. You don't have the ribosomes there. What you do have is lipid synthesis. This is where you put lipids together. And really, that's all we need to say about it. This is not cell biology. We don't have to go delving deep. Is everybody okay with that? Feel comfortable with
The first part you said, the smooth ER.
lipid synthesis? If you put fat synthesis, I'd give you credit. It's where you make the fats. It's where you put things together.
Can you talk again about the rough ER?
The rough ER, the ribosomes make the peptide chain. Inside the rough ER, that peptide chain is folded and assembled into the protein. Does that help?
So you said it makes a protein that's folded into what?
It's where the protein is folded and assembled.
Folded and assembled, you said?
Let's see if I can give you a visual. This might help. Let's pretend that this strap is a peptide chain. Okay? It's nothing fancy, it's just a straight piece of leather, right? Can I make a circle out of it? That's folding it, isn't it? Could I then twist it back on itself like that? I could now use it as a sling, couldn't I? Everybody see where I'm going with this? I could twist it around like that, pull it off. You see where I'm going? I can make a figure eight. I can do all kinds of things with just a single strand. That three-dimensional folding is what makes the protein functional. If you lose that three-dimensional folding, it kills the protein and it kills the organism. Is anybody in here familiar with an egg white? We've all seen it, right? When you crack an egg, you don't have a solid white thing fall out, do you? It's kind of a straw-colored liquid, isn't it? That's because that protein, it's called an albumin, and it's actually soluble in water. When you heat it in a hot pan or you boil it in water, you break down that three-dimensional structure. It's called denaturation. Once you do that, it's good to eat, right? Put a little salt and pepper on it, and you got yourself a nice fried egg or boiled egg. But you cannot undo that process and get a soluble protein back. Does everybody see where I'm going with this? That's where I want you to understand the three-dimensional shape of the protein is what's important. And that occurs in the rough ER. Is that clear enough for you, Micaiah? I'm trying my best to balance making sure you understand what goes on here and why it's important without going into the hardcore biochemistry and protein synthesis. That's just the place we don't need to go. Everyone's like, I agree. So is everybody with me now? And thank you, Micaiah. I appreciate you asking for clarification. Yes, ma'am.
So in the book, all those other vocabulary words, is that what you're avoiding, breaking down? Okay, I just wanna get tripped up at.
It's really, really easy to get caught up in jargon. By that I mean terms. My ex-wife used to joke that she could tell what role I was playing by what words I used. And I said, what are you talking about? She said, you scale your vocabulary to your audience. And she didn't mean it as an insult, it was actually a compliment. She said, when you're with your friends and you're relaxing, you talk like a normal person. She said, when you're in front of a class, if it is a class of seniors who are biology majors, she said, you use a fair amount of technical terms. She said, when you're talking to the other professors, I'm lost. We don't want to lose you. We're just starting out into our journey of anatomy and physiology, right? We're building that vocabulary. I would hope I can use some of these terms. I sure went to school long enough to get it, didn't I? Y'all are on that journey, so don't ever feel bad if you don't know a term or don't understand a term or you need to spell a term or something like that. It's not an insult, doesn't mean there's anything wrong. This is how you learn. So I really appreciate you. I just want to make sure that we keep our eye on the prize. We're looking at the entire body here. You need to know how the smallest unit of that body functions, don't you? You need to know what's going on inside a cell. But we do not have to make this into a hardcore biochemistry class where we're talking about rates of enzymatic mediated reactions. Nobody wants to go there, do you? Including me. I actually started off in a PhD program in biochemistry. A year later, technically, I had flunked out of the University of Tennessee. You want a story? There's a story. I flunked out of UT and got into Vandy for a full ride for a PhD. I'll tell you that sometime when we're in lab. So everybody with me? Again, thanks for asking for clarification. I'm not insulting anybody. Learn those terms if you can, build that knowledge base, but keep your eye on the big picture. We're looking at the whole thing here. Okay? So next up, we've got something called the Golgi body, the Golgi apparatus, just the Golgi, G-O-L-G-I. My old cell bio prof used to call it the thing. Because back then, we really didn't know what this thing did. The Golgi is essentially a series of membrane enclosed bubbles. It's a series of membrane enclosed bubbles. It acts basically like a packing and shipping center. A packing and shipping center. Yesterday, I ordered some blood typing kits to use with my Anatomy 2 class. I ordered them from Amazon. So Amazon gets my order, right? They send it down to the warehouse. Once in the warehouse, they have to locate blood type kits, right? Now, do we want a single kit? Do we want a 3-pack? Or since we've got a bigger class, do we want the 10-pack? Well, I ordered the 10-pack, so they have to select that size, don't they? Since I bounce between 2 different campuses and I don't know which one I'm gonna be at on one given day, I have to send it to my home address. So that means they have to put it into a package, don't they? Then they have to put the correct address on it. Then they put it on the delivery truck. Now I simplified it, but you see where I'm going with this? Your pancreas makes insulin. That insulin needs to go to a variety of cells to be functional, doesn't it? See where I'm going with this? Well, the insulin is made in the pancreas. In the Golgi of those cells, it's packaged, labeled, and then shipped out. So if you want to call it just a shipping center or a packaging and shipping center, I'm okay with that. It's a crude analogy, but I think it's accurate. I think it works. I'm okay with it. Everybody comfortable with the Golgi? Basically what it is, once it gets whatever it's intended to send out, it just forms a bubble around it, breaks off with the rest, and moves it out. Next package comes down, forms a bubble around it, breaks it off, and ships it out. Is everybody okay with that? We've also got something called the cytoskeleton. The cytoskeleton. Cyto, C-Y-T-O, is a root meaning cell. So the cellular skeleton, what does that mean? Well, the skeleton supports things. This is literally a support structure for the cell. It supports the cell, it gives it shape, it helps it move.
Can you repeat that?
It supports it, gives it shape. It's just a network of fibers and tubes. And we don't have to get any more detailed than that. Is everybody okay with that? Okay. Next up, mitochondria. Mitochondria. Mitochondria are really interesting. They probably arose as free-living bacteria that invaded a cell billions of years ago and got co-opted and captured. They actually have their own DNA. You don't have to write this down, I'm just trying to give you some. They actually can make their own proteins, but they play a critical role. And really, truly, there's only a single function that you need to know. They make ATP. They make ATP. Capital A, capital T, capital P. Just to put this into perspective, ATP is the energy source of the cell. It's the only energy source that really matters. You want to make a new protein? You need ATP. You want to break down a damaged protein? You need ATP. You want to move something into the cell? You need ATP. I used to have a good analogy for this, but technology has caught up with me, and I know nobody carries cash anymore. Am I right? We all use our phones to pay for everything. I went to get gas last week, and I saw this young woman doing this number in front of the gas pump. And she got very frustrated. And I said, "What's wrong?" She says, "It's not taking my Apple Pay," or something like that. I actually had to show her how to set it up so she could go in and pay in cash. It's kind of funny on both our ends. Because I had no idea what she's doing. I'm like, is she doing a TikTok or something? What? I gotta watch this. But if you look at a cash note, it says legal tender for all debts, public and private, right? If I gave you $20, could you use that? Go to Chick-fil-A, get me lunch, get yourself lunch. You could also say, "Too bad, Smith, I got a 20 and I need gas," and you run over here and you put $20. You can do that, couldn't you? You go make change, you give lady 10, you keep 10. See where I'm going with this? It's usable for anything. ATP is usable for anything. And so the mitochondria are where ATP synthesis happens. You got to have ATP to survive. Next up, we've got two related structures. The first one is called a lysosome, Lysosome. It's just a balloon filled with digestive enzymes. It's just a balloon filled with digestive enzymes. The second is called a peroxisome. Peroxisome. It is just a membrane-bound balloon that is a safe place to put toxins. Now, I'm kind of simplifying that, but again, it's not cell bio, we don't need to overthink this. So what happens here? Well, let's say that,
Could you repeat it?
repeat which?
The peroxisome.
It's just a safe place to stick toxin or harmful substances. Let's say that you're a white blood cell and you ingest a virus. That's your job, right? How do you keep that virus from becoming harmful? Well, you pull it over to a lysosome. Those digestive enzymes are in that bag, you stick the virus in the bag with the digestive enzymes, it breaks it down. Everybody go with that? You get a protein that you're no longer using, you stick it in the bag, you break it down. Everybody got an idea of what a lysosome does? Peroxisome, let's say that you get some kind of toxin into the cell. You put it into a peroxisome where it can do no damage. Eventually, it will be removed from the cell or broken down. Everybody okay with that? Next up, the cytoplasm, C-Y-T-O-P-L-A-S-M, the cytoplasm. Cytoplasm is basically the liquid that fills up the cell. If we were to make a model of a cell, we'd really kind of make like a water balloon. But instead of water, we would have cytoplasm. Cytoplasm is thicker than water. It's not like jelly or Jell-O, but it is not exactly like water either. It's kind of more like a light oil in just in terms of how it flows, or pancake syrup. You get the idea? Anybody here ever spilled pancake syrup? It's a real mess to clean up, isn't it? It gets sticky. You ever spilled soda and come back a day or two later, it's dried up? It's sticky, isn't it? Well, that's because of the dissolved sugars that are in the syrup or the Coke or honey. We've all had honey, it's thick, right? You get an idea of what cytoplasm is, kind of like those three. Cytoplasm, number one, allows transport of substances within the cell. It allows for transport of substances within the cell. So for example, I need to move an RNA molecule from the nucleus to a ribosome. It travels through the cytoplasm. Everybody see that? The second thing is it provides a location for reactions to occur. There's a lot of different things dissolved in the cytoplasm. When those molecules collide with each other, they can react. The third, it gives shape and support. We've all blown up a balloon, right? Has anybody here ever made a water balloon? You put water in, it gives shape to the balloon, doesn't it? See where I'm going with this? Without something to fill it up, the cell would essentially deflate. We've all seen plants that wilt when you don't water them, right? What's happening is the water is leaking out of the cell and they essentially deflate. And that's the cytoplasm. Questions? Everybody stand up, stretch, pop your joints, take a 30-second break. Y'all are starting to give me that 1,000-yard stare. I mean it, I'm serious. Do something, move, stretch. There's a reason I get up and walk around. If I sat here for this long talking about nothing but organelles, I'd put myself to sleep. Okay, everybody a little more awake, feel a little bit better about things? What do we got, about 2 pages of notes, 2.5? Okay. Next up, the cell membrane or the plasma membrane. A lot of people don't really understand the role that the plasma membrane place. Some people would say protection, shape, things like that, but really what it is, it's a control mechanism.
Can you repeat that?
It's a control mechanism. It's a gate. Could I control who comes in to my classroom? I could lock that door, couldn't I? Several of you got here late. I could decide if I wanted to let you in, couldn't I? You see how I'm controlling access? That is what the plasma membrane does. Certain things you want to come into the cell. You want sugar to come into the cell because you need energy, don't you? You want oxygen to come into the cell because you need that to break down the sugar. You want water to come in. But you don't want so much water that you pop the cell. Everybody see where I'm going with this? You don't want to lose things from the cell. I don't want y'all leaving until we're done with the lecture because you might miss something important, right? No, I'm not going to lock the door and tell you you can't go. This is class, not a prison. But you see where I'm going with that. It acts as a gateway. It is a gatekeeper. And you definitely want that gatekeeper. You want to control what goes in and out of the cell. Now you've got three types of transport at work here. The first is called passive transport. Passive transport. A simpler name for it would be diffusion. Diffusion. Let's say that I had a jar full of some very foul-smelling substance, and I opened that jar and set it right here. ZZ and Taylor would be the ones to smell it first, wouldn't they? Because they're the closest. Aiden, Chrislyn would probably be the last, wouldn't you? But eventually, everybody is going to get a nice whiff of whatever nasty substance I opened up. The reason for that is called diffusion. Diffusion means a substance moves from an area of high concentration to an area of low concentration. High to low. If I park my car on a hill and I leave it in neutral, what's gonna happen to it? It's gonna roll down, isn't it? I'm not gonna have to push it, it's gonna go on its own. We all agree on that? That is how passive transport works. It's called passive because the cell doesn't have to spend energy. The cell does not have to spend energy. Now, if I parked that car at the bottom of the hill, is it going to get its way to the top? We're going to have to expend energy, aren't we? I'm going to have to offer 10 points on a final to every student willing to push that car up the hill, right? I'd be willing to bet I can get some helpers. If I offered you 10 points in the final and 3 slices of pizza, I'd get even more helpers, wouldn't I? But you're gonna have a hard road to hoe pushing that thing up, aren't you? Y'all know what kind of car I drive. Maybe you're gonna be a Chevy Avalanche or something. No, it's a little SUV. That's active transport. You're going against a concentration gradient. You're going up a hill. If I had that foul-smelling substance and I were to gather it together and put it into the jar, I'm concentrating it, right? But I'm having to expend energy to do that. Active transport. And then The final form is called semi-active transport. Semi-active transport. Now, what do I mean by that? Does this pen want to fall to the table? Well, right? Didn't cost me energy, did it? Now, when I bend over, if I can, pick it up, I had to expend energy, didn't I? Passive and active. Look at how I'm holding the pen. I've got it cradled in my interlock fingers here. Does everybody agree? Does it want to fall to the table? Okay. Yeah, it does. It's a higher energy state where I'm holding it, isn't it? But there's a barrier there. Does everyone see that? Am I releasing that barrier, opening it? Everybody saw how I opened my fingers and it dropped. Did I expend energy? What do you think?
Yeah, because you moved your feet.
Yes, I had to move my fingers, didn't I? I had to open the gate. It wanted to come through, that's passive. The actual movement was passive, wasn't it? But I expended energy to open the gate, so that was active, wasn't it? Semi-active requires energy to open the gate. Semi-active requires energy to open the gate. And again, it's probably a gross oversimplification of all of this, but I think everybody's comfortable with it. Am I right? We're not done. Lydia's going, dang, I thought we were.
I got, the passive and then I got semi-active. What was the third one?
Semi-active.
I got that.
You got
Missing
passive,
active.
active, semi-active.
Can you go over active again, please?
Active is going against the gradient. Let's say I've got a bottle of syrup. If I tip that bottle of syrup over, is it going to pour out? Doesn't require any effort at all, does it? Gravity pulls it, it's going to fall. If I were to scoop that syrup off of the table and put it back in the bottle, am I concentrating it? I'm going from a low concentration back to a high concentration, right?
Yeah.
Does that require energy?
Yes.
Yeah, because I'm scooping, right? Everybody see that? That's active. Visualize it as a hill. Which is higher energy state? This hand right here, Micaiah? Or this hand down here?
What was the question again? I'm sorry, I should have wrote that.
Which is a higher energy state, up here or down here?
Which would require more energy?
Higher energy state. Which is higher? Yeah, everybody agree? Because if I release something here, it's going to fall farther, isn't it? That wants to happen. It wants to go down the hill. It wants to go down the gradient. It wants to move from high to low. We all agree on that, right? That's why we don't park our car in a hill and leave it neutral. Because we don't want the car rolling back down the hill and crashing into something, right? Now, if we park on a level piece of ground, perfectly level, can we leave our car in neutral? May not be a good idea because somebody could always come along and push it, right? But as long as no force acts on it, is it going to stay where it is? Yes. You see that, Micaiah? There's no transport in that case, is there? Now, if I want to get it back up to the hill, I've got two choices. I get it and start it, burn gas to expend energy to move it against the energy. Do you see what I'm doing? Or I get a bunch of y'all and you have to expend energy to push it up that hill. And once we get it back up there, it's at the higher energy state again, isn't it? Can it roll back down? See where I'm going with this? This is why we have to always acquire energy from our environment. We always have to eat. Okay? Is that clear, Micaiah?
Yes.
All right, the last thing I want you to get is the extracellular matrix. But we're just going to call it the ECM. ECM. The ECM is a network of molecules that extend out from the plasma membrane and beyond the cell. They extend out and beyond. Essentially, they act like glue. They hold the cells together. If you need to text, please do so outside of class. Thank you. They also allow communication between cells. An example of that, blood is mostly red blood cells, right? You've got a few white blood cells mixed in amongst it. Those white cells are immune cells. They're looking for people who don't belong. They're looking for things that are foreign. How do they know something is foreign? If a white blood cell bumps into a red blood cell, the molecules on the ECM of each other interact. It recognizes that as belonging, and it moves on. Does everybody see that? If it bumps into a bacterial cell, that's not the same, is it? It's different. Boom, gets chomped. Is everybody clear on that? Thumbs up high like this if you are. High. Yes, Micaiah, that includes you. So you don't get it. Okay, everybody got it.
Can you, what did they hold together? What did they hold together?
The cells.
Okay.
I mean, think of it as water balloons. If we said the cell's like a water balloon, you don't get water balloons stick together, do you? If you smear both of them with super glue, you've now got a big balloon, don't you? See how it works? The ECM also can be made stronger and protective. Bone. Bone has an ECM that is very rich in minerals like calcium. Bone is hard, isn't it? Because of the ECM. The ECM, believe it or not, can also be entirely liquid. Can anybody give me an example of an ECM that is completely liquid? Blood. Blood. It can be rich in protein. Things like cartilage It can be rich in fats. You get an idea here. And one thing I want to make sure that you understand is what we have talked about today is a generalized human cell. Most cells in humans are very, very specialized in their function. But since we are out of time, we will talk about that on Wednesday, or next Monday. Everybody clear? Everybody cool? Questions, thoughts, gripes, complaints?
It stands for the extracellular what? The ECM stands for what again?
Extracellular matrix.